right, so we are now being recorded, and I will begin my presentation for this video teleconference. Okay, so as usual, let me begin by asking you questions and let me hear from you. Okay, you don't have to answer this question per se, but you can say something aside from maybe your answer to this question. Okay, but I would like to be able to hear from you on how were the quiz and the exam, or maybe the group work that you've just made, group work number 1. So can I have some volunteers? Maybe there are some concerns on the quiz and the exam. So I want to hear from you. No one? Do you think you were prepared for the exam? Okay. Anyone from the group want to answer that? You, uh, did you feel prepared, or was it too sudden and very little information were given during the lectures, and then you were sort of shocked during the exam, the quiz? May I hear from just a representative? I'm gonna call names. Is it okay to call names? Okay, may I hear from Rangel Galileo?
Hello, sir. Hello.
Yes. Okay. So I have here a question. How were the quiz and the exam?
Um, for the quiz, sir, I think I did okay. For the exam, I believe I could have done better than what I've done, sir.
All right. Thank you for being honest. How about accessing it, or were the contents or were the type of quiz and exam okay, in your opinion?
Um, type of quiz. I guess, sir, it's okay, multiple choice and stuff.
All right. Okay, thank you. What about the others? Are there comments before I say something? No comments on the quiz and the exam? Um, the quiz is multiple choice, or was multiple choice, right? I don't know if you've encountered that type of multiple choice wherein you have to choose several answers for one item. So I hope you were able to do that successfully. And in the future quizzes, that type of question will still come out. Okay, multiple choice, but there are multiple answers as well in the choice that you have to select all of them in order to get the full point for the question. So, and the exam, I think, is very straightforward, multiple choice. And I hope the time given for the quiz and the exam were adequate enough for you to finish. I've noticed some of you just took half of the time and finished the quiz and the exam. That's fine. And there are also some of you who utilized the whole hour for the exam and the whole, I guess, 15 minutes for the quiz. Okay, so are there any concerns or comments? Again, your last last opportunity to ask questions or comment. With regards to the answer to the quiz and the exams, I know I haven't revealed the answers to you. And for now, I am intending not to. Okay? But I know you know your scores. Okay? I hope you've seen your scores. But right now, I'm not intending to reveal yet the answers for the exam and the quiz. But you know your mistakes. So I think let's keep it that way so that you will think over it and go back to your notes or book in order to find out what the correct answer really was. Okay, so that's what I'm planning for the answers to the quiz and the exam. So you only know your scores, but not the correct answers. All right. This is our schedule. We are now on the 20th of January and we are doing our third video teleconference. And our lesson is supposed to be for chapter 3. Um, chapter 3 is going to be spread for 2 weeks. Okay, so there's Mendelian, it's about Mendelian genetics. For this week, we are going to set the foundations, okay, or all of the principles, the laws in Mendelian genetics. For next week, I don't think I will be posting new lessons because it's still the same chapter and I think I posted all of the lessons already within this week. Instead, for next week, it will just be an additional homework for you by pair, still on Mendelian genetics. Okay, so that's the topic that is currently active in eLearn right now, and I hope you've started viewing the videos. There were 3 videos posted, and Mendelian genetics is sort of going to need you to recall your basic probability. Okay, so it's like solving a simple puzzle. That's how Mendel actually got to establish the laws. Not if you already listened to the videos. So it's gonna be more. It's gonna be less wordy. It's gonna be more on analyzing and solving puzzles, if you want to call it a puzzle, or computing simple probabilities like the multiplication law of probability or the addition law of probability. So that's for this week, and next week that's Mendelian genetics. All right. For the rest of this video teleconference, I would like to review all of us on our lesson from the last weeks, okay, especially the cell cycle. Okay, so let me call on Nisel Bakayana. Can you say something about the G1 phase of the cell cycle? Naba si Nisel? Okay, anything at all? It doesn't have to be the exact words that the book presents. But what can you remember about the G1 phase of the cell cycle, Miss Bakayana? Wala? Okay, I'll go back to you, okay, later. Let me hear from Mr. Bakonga.
Sir?
What can you say about the G1 phase of the cell cycle as shown in this illustration?
Again, sir? Chappy, sir.
Say something about the G1 phase of the cell cycle.
Kana nasa on the screen, sir.
Yes.
Kon, sir. Muna siya ang cell division nga ma sa mitosis then and the beginning of DNA replication during S phase. Di ra magsugod ang sir. Makipay ang mga cell division, sir. At tapos pagguman din, sir, kay magadto din sa S phase.
Okay. The G1 phase, yes, you're correct. It's before the S phase. The G1 phase is part of interphase. Okay. This is the portion when the cell seemingly is not doing anything. That's the interphase. but it's actually preparing. Okay, so in the G1 phase, there is the checkpoint for any DNA damage, okay, that the cell has incurred during mitosis or maybe during mutation, when it was exposed to mutagens such as ultraviolet rays from the sun. So the cell actively checks if there are damages in its genetic material during G1 phase. Okay, so and when a damage is found, the cell will try its best to repair the damage. So that's what's going to happen in the G1 phase. And then there is a checkpoint, and the checkpoint will stop the cell in the G1 phase if the damage is not repaired. Okay, so that's okay. So there is also this phase called G0 or GO phase. Okay. Um, Let me call on Miss Hala. Okay. Say something about the G0 phase of interphase.
Hello, sir.
Yes.
Okay. In sa G0 phase, cells that enter in that phase, I guess, remain viable and metabolically active but are not proliferative. Like, when terpod ang cancer cells or sa ako na-remember sa pagbasa kay some of them will just pass through it and other cells kanang muna sila ang katoy mong gingon sir nga mga dead cells dito
Those that have mutation that cannot be repaired.
o cannot be repaired sir dira sila mo enter then mubalik dayon sa G1 para i-proceed ang process.
All right. Thank you. Thank you for your sharing. I think you really remembered what you've read from the book, no? Okay, so the G0 phase is for cells that will no longer be entering the mitosis or meiosis portion of the cell cycle. You're correct there. So cancer cells skip this phase. You're correct also there. And certain cells undergo G0 stage or phase if there is a mutation and it cannot be repaired. So they will stay at G0, non-dividing. And specialized cells like muscle cells or neurons, because they are so specialized already, they cannot anymore undergo mitosis. So they just stay under G0 phase. But I like what you mentioned a while ago. I would like to reiterate it that even if the cell is not dividing anymore, it's still metabolically active at G0 phase. So it's still performing its work. Okay. In fact, it's already matured and it's doing its work efficiently when it's in G0 phase. Okay, if it's a muscle cell or a nerve cell. So thank you very much for that. So if the G1 phase is successful, meaning to say the genetic material is not mutated or has been repaired, the cell will then undergo S phase. Okay, may I ask Mr. Rejos, okay, what is happening during the S phase, or can you say something about S phase?
Sir, what I remember sa S phase is that the DNA will start to replicate here, sir, and also the centrosomes food, sir, that will help separate the DNA, sir, later in mitosis.
All right, thank you. So there is a replication of genetic material here in preparation for cell division later on during mitosis or meiosis. Okay, so the genetic material, specifically DNA, will double at the end of S phase. Okay, for the centrosome though, the centrosome will only divide at around prophase already. Okay, not yet so in the S phase. So in the S phase, expect at the end of S phase, double the amount of genetic material. All right. So thank you for that. So again, that's logical, right? Because you need to make sure first that what you're copying is correct. That's the purpose of G1. Then S phase is now the photocopying. Okay, you don't just want to photocopy a book, then all the pages are scrambled. Okay, you want it first to be filed properly according to page, and that's G1. And then S phase is the act of copying already. Okay, so I hope that is simple enough to understand. Next, in the interface, we have the G2 phase. Okay, may I ask Mr. Coruña, can you say
Yes, sir.
something on
It,
the G2 phase?
yes, sir. In G2, the size of the cell has roughly doubled, especially in the nucleus, and its DNA has already been replicated, and it is already ready for, most likely ready for mitosis, sir.
Yes. Okay. Thank you for that. There is also a checkpoint in G2. Okay, but it is not as strict as the checkpoint in G1. Okay, so there's a checkpoint in G2. The checkpoint is all on the size of the cell, as you've mentioned. Is the cell big enough to undergo cell division? So if not, then it needs to continue still in G2 to continue growing. Okay, so not only is the size of the cell increasing, but also some organelles are getting replicated during G2. So for example, more mitochondria might be made, okay, in preparation for cell division, or more chloroplast might be made, okay, by the cell. Okay, so thank you very much for that. Now, after interphase, which is the longest stage of the cell cycle, as you can see in this illustration, there is that cell division stage. So in this case, it's mitosis. And it's composed of, we're familiar with this, the phases: prophase, prometaphase, metaphase, anaphase, and telophase. Okay, so in mitosis, there are two, we can classify the events in mitosis into two. First, we have cytokinesis, and then second, we have karyokinesis. Okay, so can anyone from the group tell us the difference between cytokinesis and karyokinesis? Any volunteer?
Sir, if I may, sir.
Yes.
Basically, karyokinesis is the division of the nucleus or the nuclear material, and cytokinesis is the division of the cytoplasm itself or the cell itself.
Yes, okay, that's correct. Karyokinesis is usually very evident during anaphase, right? When the sister chromatids in mitosis separate, so the genetic material are separated from each other, and that's karyokinesis. Cytokinesis will only begin to happen at telophase. No? In the case of animal cells or cells without cell walls, a cleavage furrow will form to facilitate cytokinesis. And in plant cells, a cell plate is going to be deposited in between the new daughter cells for cytokinesis. All right. So that's a summary of the cell cycle. Let me proceed. So here are some illustrations. This is for mitosis. Okay. So just a note here that during interphase, during the G1 or G0 or the S or the G2 phases of interphase, the chromosome will remain to be a chromatin in structure, okay? Chromatin in structure, meaning to say it's uncoiled and it's sort of messy throughout the interphase, okay? So again, during the G1, G0, or S, or G2 phases. So sometimes it's very difficult to see have the DNA duplicated or replicated already. It's very difficult to look at the evidence of DNA replication, not unless the cell is already in prophase and the later stages of mitosis. So in prophase, the nucleus, the nuclear membrane disintegrates, starts to disintegrate, and the chromatin, okay, which are the chromosomes, start to condense. And then here you can see the chromosome. So I just like to note in this illustration, this one thing here that I'm encircling, this is one chromosome. Okay, so the illustration makes it appear like it's a homologous pair that's pairing together. No, this is one chromosome, because if you look at the prometaphase here, there are only 4 chromosomes. So this is one chromosome, and then this one is another chromosome. And you can now see the rabbit structure. It's sort of like a rabbit ear, and then pinched in the middle. And the reason is there are now sister chromatids because of DNA replication per chromosome. Okay, so that's in prophase. And then, as mentioned a while ago by one of your classmates, the centriole, okay, is going to move in opposite poles and start to divide also during prophase. All right. Then in prometaphase, it's now obvious, okay, that we have a replicated genetic material here, because one chromosome is made of 2 chromatids already. So the chromatids are identical. So question, this is for anyone in the group. How many, what is the diploid number of the cell here in illustration? What is the diploid number?
4, sir.
All right, there are 4 chromosomes here. So we have 1, the first one. This is 2, 3, and then 4 chromosomes. So that's a diploid number. How many chromosomes are from the mother? Anyone?
Also 4, sir.
4?
Oh, wait.
Remember, diploid is a combination of mother.
2, sir.
Okay, so
2,
if there
sir.
are, if the diploid is 4, if that's a diploid number, there are 2 from the mother and 2 from the father, color coded here as red and blue. Okay, so look at this cell during prometaphase. Okay, question: how many sister chromatids are present?
Eight, sir. 8.
Yes, okay. Each chromosome, because the genetic material has been replicated, will now have 2 sister chromatids each. So 2 times 4 chromosomes, that's 8 chromatids, correct? Another question, how many centromeres are present here in prometaphase? Centromeres.
4, sir.
4, yes. There are only 4 centromeres here. So this is 1, this is 2, and of course you have 3, and then 4 centromeres. Okay, the centromere is a region where the kinetochore is found, and the kinetochore is the portion where the spindle fibers attach. So you can see here that the spindle fibers are also forming already. You can see these lines, okay, and they will attach to the kinetochore of the sister chromatids. And here we can see in the metaphase stage that there is an imaginary plane called the metaphase plate, where the chromosomes align, okay, in the plane. So remember, this is mitosis. There is also a procedure that scientists do to these metaphase chromosomes in order to make this. Okay? Question to anyone, how do we call this diagram? The one here on the right, how do we call this?
Karyotype, sir.
All right, that's the karyotype. You can also call it a karyogram, okay? And it shows all of the chromosomes, in this case, diploid number, chromosomes, in this case, human being, okay, that are arranged according to homologous pairs, okay? And homologous pairs are recognized based on the shape and the location of the centromere of the chromosomes. Notice here, all of the chromosomes are duplicated, currently duplicated. You can see here, for example, it's like a rabbit shape, Okay, there's one chromatid for this side, and then another chromatid for this side. Okay, so it's replicated. And if I may change my ink color, that's the mother chromosome. That's from the mother. This one is from the father. This blue one here that I'm drawing, and it's also replicated. Okay, so this is a diploid cell. Okay, composed of it contains the diploid number 46 chromosomes. All right. But question for anyone in the group: how do scientists get the chromosomes out from the cell? How do scientists get the chromosome from the metaphase stage cell here, the one that I'm encircling? So these are the chromosomes that are going to be made into a karyotype. No, their pictures will be taken and be made into karyotype. How are they taken out of the cell? Or how is mitosis stopped? Have you researched on that? Have you read on it? Okay, perhaps you haven't gone through it, or maybe you've forgotten about it, but let me tell you how. Okay, take note, the driving force of the movement of the chromosome will be the spindle fibers. Okay, so those are the, let me go back here. Those are the microtubules emanating here from the centriole. Okay, so this is the driving force. So what scientists do in order to stop the cell division at metaphase is they try to disrupt the formation of the spindle fibers. So the spindle fibers will not form successfully. They will use a chemical called colchicine. Okay, colchicine. So take note of that. Colchicine to stop the formation of spindle fibers, and therefore the chromosomes will not be moved by the spindle fibers, and they can just break the cell, and then they can, as we see here on the left, there's a disarranged array of duplicated chromosomes, and then they use scissors, they cut the pictures and arrange the chromosome by pair, as you can see here, based on size and centromere position. So that's how they make the karyotype. They use a chemical called colchicine to disrupt the formation of spindle fibers. And then they break the cell and remove the duplicated chromosomes, take a picture of them, cut the picture, and rearrange them according to homologous pairs. And if we do not stop the cell division at metaphase, it will continue to anaphase. So I have a question again for the group. How many chromosomes are present now in anaphase here? Anyone?
24, sir.
Look at the illustration. Remember, we started with 2n = 4. Okay, so how many chromosomes now are here in anaphase?
Try lang, sir.
All right.
There are 8 daughter chromosomes, sir.
All right,
Yes,
there will now be 8, okay, because the sister chromatids are now separated from each other. So a while ago, when they were still together, each one of this rabbit-shaped structure is considered as one, okay? But during anaphase, when the sister chromatids are separated, okay, they now become separate. So they can be counted as 2, okay? So from 4 to n, now during anaphase, because the cell has not completely divided yet, there are 8 chromosomes in this cell right now, okay? And then during telophase, okay, during telophase here, because the cells are now about to divide or separate in cytokinesis, okay? Well, this one here in anaphase is karyokinesis. This one is cytokinesis, Okay, there are now 4 chromosomes for this daughter cell and 4 chromosomes for this daughter cell, and they are identical cells to each other and to their original parent cell. Okay, so that's mitosis, and we can see here in plant cells there's a formation of cell plate instead of a cleavage furrow. Okay, so I think those questions were part of your seat work, right? Okay, so we will talk about the seat work soon. So before the sister chromatids are separated, though, they are connected to each other with a molecule called cohesin. Okay, and the cohesin is sort of protected here in the centromere region by another protein called sugoshin, which prevents separase, an enzyme, from degrading cohesin. So everything here will be degraded on this portion and on this portion, but the ones in the centromere region stay intact. So the chromatids attach at the centromere region and detach on either ends of the chromosome. All right, so this is the structure. So again, sister chromatids are identical to each other, okay? Base per base of the DNA that they contain. Here's a comparison of mitosis and meiosis. Okay, any volunteer who would tell us the events that happen in prophase 1 of meiosis? It might be not in order, but any event at all that happens in prophase 1 in meiosis. No one? Okay, we can see here. Okay, continue. Sorry.
Over, sir.
Crossing over, yes, that's correct. Crossing over will happen, okay, between homologous chromosomes, okay? And crossing over is achieved first by a process called synapsis, okay? So synapsis. So as you can see here, the homologous pairs will pair up, okay? They will pair up in meiosis 1, prophase 1, but in mitosis prophase, they will just randomly group themselves. But in meiosis 1, prophase 1, okay, they will really pair up and they will undergo synapsis, where the formation of the overlap called the chiasmata will be observed, and there is crossing over when they will be exchanging materials, okay? The homologous pair from the mother will exchange materials with the homologous pair from the father, for example. And that's why we have here what we call as a tetrad, okay? The tetrad will just be the homologous pairs, and each of the sister chromatids are obvious already. And because there are 4 sister chromatids per tetrad, that's how they appear, so we call it a tetrad, okay? We also have the term bivalent. And the bivalent is no different from the tetrad. Okay, a tetrad is essentially a bivalent. But in a bivalent, the sister chromatids are not so obvious, so they don't appear to be 4 sister chromatids in a bivalent. All right, and then here we have meiosis. Yeah, this is meiosis 1. So we can now see here that after meiosis 1, the resulting daughter cells will now be haploid. So from 2n, originally, after meiosis 1, there will just be n number of chromosomes. So from 4 here, meiosis 1, beginning, after meiosis 1, there are only 2 chromosomes per daughter cell. I'm sorry for that. Okay, 2 chromosomes per daughter cell. And then after meiosis 1, the cell will undergo meiosis 2, and the sister chromatids will now be separated so that there will be at most 4 new daughter cells from one parent cell, and each daughter cell will contain 2 chromosomes, and each chromosome only has 1 chromatid, as you can see here in this illustration. So 2 chromatids, 2 chromatids, 2 chromatids, two chromatids. So the result is haploid. Okay? And meiosis is the process that's used in gametogenesis, in the production of sperm cells in spermatogenesis, and the egg cell in oogenesis. Okay? So this is a summary of spermatogenesis and oogenesis. And just notice here in spermatogenesis, one spermatogonium, which is diploid, will be able to produce 4 new sperm cells here at the bottom. But in the case of oogenesis, one oogonium will only be able to produce 1 ovum or egg cell. Okay? So 1:1. This is a question from your seat work, if you can remember, or from your homework. Because there is an unequal cytokinesis happening in oogenesis. So there's the formation of polar bodies. There are 2 polar bodies formed. And these are sort of useless already because a majority of the cytoplasm are given to just the other half, no, of the division. So only 1 egg cell will be produced from 1 oogonium. But 4 sperm cells will be produced from 1 spermatogonium. Right, so I'm not sure what's happening here, but let me erase some of these stuff over there. Okay, so this is the problem set. And I hope you got your scores already. Okay, I already posted your scores. And I posted the checked work that you've submitted to me. I've uploaded them back to you, okay, in eLearn. So maybe we'll not answer these anymore right now because you already know the answers, unless you have a question that you want to address to the group. Are there any questions on this portion of the problem set? Okay, there were some groups whom I think had a challenge really on counting of the chromosomes and chromatids and centromeres. So I really hope that the corrections or the answers that I've given you have enlightened you and that you understood why those were the answers, okay? And again, if you don't understand, please ask questions. So here's the other part of the homework. Okay, the red blood cell of humans do not have nucleus or nuclei, so there are no chromosomes in red blood cells. Okay, but in other animals like amphibians, the red blood cells have nuclei. So unique yung sa humans, no? We don't have, our red blood cells don't have nuclei. Okay, any other points of interest here? So look at this, number 4, letter B. How many sperms will be produced from 200 spermatids? So spermatids will mature into sperm, 1:1. So 200 spermatids will result to 200 new sperm cells in a process called spermiogenesis, or the maturation of sperm cells from spermatids. Letter C, how many eggs will be obtained from 200 primary spermatocytes? As I mentioned a while ago, for egg cells, it's 1:1. Okay? But for sperm cells, it's 1 primary spermatocyte and 4 sperm cells. But in egg cells, the answer in letter C is still 200. Okay? Because the rest will be polar bodies. Then we have this third portion. Number 5 here, this question on plants will be very much repeated in your genetics laboratory activity. I think that's lab activity number 4. So I hope you will study well the life cycle of the plants because it's much more complicated than animals' life cycle. Okay, there is the involvement of gametophyte and sporophyte in the plant life cycle. So are there questions in this portion of the homework? I hope you can recall that in flowering plants, there is such thing as double fertilization, okay? Meaning to say, two sperm cells will enter the ovary of a flowering plant. And one of them will fertilize the egg cell, the ovum, and the other one will fertilize the endosperm, okay? The endosperm is a triploid. So it will have 3 sets of the haploid number, okay? So in this case, number 5, endosperm cell will have 72 chromosomes because it's triploid, while the egg cell will retain the diploid number, 48. That's double fertilization in flowering plants. And lastly, we have this question on hybrids, the mule, okay? And I think all of you got the answer here correctly, that the mule will be sterile because there is one pair of chromosome, or there's one chromosome that doesn't have a pair, because the mule is a result of a hybrid of a horse, which only has 30 pairs, and an ass, which has 33 pairs of chromosomes. So there are, sorry, not one pair, 3 pairs, okay, will not have, 3 chromosomes will not have a pair if the horse and the ass are going to form a zygote called the mule. So the mule will be sterile. It cannot produce viable sperm or egg cells. Last but not the least, I just want to remind you, we're now in chapter 3. So I hope you are almost done reading chapter 3 on Mendelian genetics. Or you can watch the videos that I posted. I already posted 3 videos. Okay. And then there is a homework number 2 that's posted, that's due this Saturday. Okay. And for next week, we are still going to be on chapter 3. So you have 2 weeks to dwell on chapter 3. Still on Mendelian genetics next week, but it will be more on additional problems on Mendelian genetics for next week. So group work will be due on January 23. I'm sorry. There is also a quiz on January 23 about a portion on Mendelian genetics, okay, based on the group work also. All right, so with that, are there any questions or concerns? May I hear from you? Questions, concerns?
Chapter 3 to sir, no quiz.
Yes, chapter 3 is Mendelian genetics. The lesson right now in eLearn is chapter 3, Mendelian genetics. So this quiz though is not is based on the group work, okay? Because Mendelian genetics is going to be divided into 2 parts, okay? Okay, there is no clear cut though between which part will be the boundary. But I suggest you really finish reading chapter 3 this week, so that next week when we talk about it again, you will understand it more because you've already read the whole chapter. So that's chapter 3, Mendelian genetics. I noticed that you keep in schedule, no, with regards to checking the lessons in eLearn for our course, because our schedule is Wednesday, Saturday. I understand that I think all of you were not able to check new lessons last Monday. So for example, when I checked the people who have viewed the videos, there were only views, I think, it started yesterday, late yesterday. But I suggest to you that even if it's not our class time, say Monday, you already start viewing the videos or reading the chapter. So don't just wait for Wednesday or Saturday in order for you to do your genetics activity. You can still use the time in the other days of the week to study the course. Not if you're free, if it's your free time. So I'm not telling you to abandon your other subjects. I'm just telling you have the option to study genetics throughout the week, not just on Wednesdays and Saturdays. Especially that our topic is getting a bit more complicated. Okay, so Mendelian genetics might require you to read and reread and to practice solving. In the videos that I posted, there are problems that I solved in the video with solutions, but I did not solve all of the problems. I left a majority of them for you to solve. Okay, the problems found in the book. All right, so any other concerns, questions? None so far. I guess there's none. Okay, so what can I say? First and foremost, we are done with cell division, mitosis and meiosis, okay? But you have an activity in your laboratory that would still involve mitosis and meiosis, especially on life cycles, that's meiosis. So you really need to pay attention also in your lab activity, okay? Because there are portions there that might not be included in this lecture, especially now with the time constraint, and a majority of the work will be yours to read your book. In my lectures that I posted in eLearn, there are, of course, certain points, especially in meiosis, that are not fully discussed. But unless you ask me about it, I can answer you. So in your laboratory portion of this, it's, I think, more detailed, especially on the life cycle portion, which I believe you're also doing right now. So if there are no questions, and if you have questions later on, just post them in eLearn or in the Messenger, and I will answer them right away. So for now, I would like you to spend time on reading chapter 3 and spend time with your pair to analyze and answer group work number 2, which is now posted in eLearn. And the due dates for these will be on January 23, 2021, this Saturday. All right. So again, concerns, just ask questions. So if you don't have any more questions in this video teleconference, I thank you very much for joining. Okay. And the recording of this video teleconference will be posted later on in our eLearn wall.